Are there any questions? All right, hopefully you can see my screen. Just want to go over a little bit with the syllabus. I will be in class from 6:30 till about 8 o'clock. And I will, after I talk about the lab, and obviously I'm going to talk a little bit about the syllabus today, I will just stay here and answer questions, sort of like in the lab. This is the same syllabus that we went through before. Let me just talk about a few things that are particular for the lab. Here's something I didn't mention, and that is the quizzes will have both lecture and lab questions, and that includes the final exam, so you will be quizzed on some material you have in the lab. And there will only be, well, quiz 1A and quiz 1B will cover the first couple of labs. There are some worksheets that are due in the lab, and they're going to be worth a total of 50 points, and generally they will be due Saturday during the week that that lab runs. I talked about microscopes. There's something I needed to talk about. Oh, the safety information for students and physicians. I'm going to have you read through it. It's not as important in an online class because you won't be working with microorganisms. It was more important when you were actually handling microorganisms. You do need to know the laboratory safety and rules. You will be quizzed on them. Safety information for students and physicians, I don't think you're going to be quizzed on that, but you will be quizzed on the laboratory safety and rules, and that's because we feel that you're taking microbiology, you should know how to handle microorganisms appropriately and stay safe, even though you're not actually going to be handling microorganisms. So read through these rules. You are required to follow all of the rules, and you could be tested on any of them. Let me go over a few that will be really important, the most important ones. When you come into the lab normally, you put your backpack and your books and your coat in a cubby hole, and that is not considered part of the lab. You then walk towards the benches and the table, and immediately when you leave the site where the door is and the cubby holes, we consider that inside the lab. And anything that comes into the lab has to stay in the lab, and you cannot take it out of the lab unless it is autoclaved or put in bleach. So if you, for example, you were to go and start using your phone in the lab, it would be confiscated and you wouldn't get it back until it had been soaked in bleach or put in the autoclave. If you don't know what an autoclave is, it's like a giant pressure cooker. And obviously both of these treatments would kill microorganisms that may have gotten on your phone, but they would also destroy the phone. So anything that comes into the lab never leaves the lab until it has been autoclaved or put into bleach. Now you could bring in papers, for example, and at the end of the term they could be autoclaved or bleached and then given back to you, and then you can take them home. But generally things that come into the lab stay in the lab, and things that are in the lab stay there and don't leave the lab. Now, as I stated, with your books and your jacket and your backpack, those go in the cubby hole, and that's not considered part of the lab. It's considered outside the lab. Any question about any of that? The reason for that rule is to keep the microorganisms that are in the lab from leaving the lab. There actually was a case which happened in Clark College where someone was working with, I think it was a species of Salmonella, and they took this species home with them and then made a meal for their significant other, and their significant other came down with a Salmonella infection, which they were able to prove came from Clark College. And personally, I would say that student wasn't handling material appropriately in the lab, because if she had been, she would not have taken the bacteria home with her. And we have these rules in place to prevent things like that from happening again. So everything that goes into the lab stays there unless it's treated or autoclaved. Any questions about that? No eating or drinking or smoking or chewing of gum in the lab at any time. Students must wear a lab coat and nitrile gloves, and you take off the gloves when you leave. You wash your hands. Well, actually, you take off the gloves, you take off your lab coat. The lab coat you just hang up. It is autoclaved in between terms, but it's never autoclaved during the term. And you then wash your hands, and then you leave. And coming into the lab, you always wash your hands. Well, you put away your books and stuff like that. And then you go wash your hands. You dry your hands. You put on your lab coat. You put on your glove. And then you go to your position at the bench. So gloves and lab coats are required all the time in the lab, and that's to prevent bacteria from spreading. Whenever you're working with a splash hazard or a chemical hazard that could get in your eyes, goggles must be worn. Before you begin, so usually at the start of the day, you disinfect your lab bench beforehand with disinfectant, and then when you're all done, before you leave, you also disinfect the lab bench. So you do that always before you begin and after you're done. Report all accidents to instructors. There is a procedure for wiping it up, but you won't be in the lab, so I'm not going to go over it. You can read about it. I won't go over that. All cultures must be labeled completely with your name, the date, the nature of the specimen, the medium used, and then it's optional. You can put the temperature that you're growing the culture at. The culture is the microorganism you're growing. So name, date, nature of specimen, and medium used are always required. Cultures and other material you're done with must be discarded properly. If the material is not contaminated with bacteria or a microorganism, it can be put in the trash can, and that includes all paper and plastic disposables that do not have bacteria on them. No glass ever goes in the trash can. Glass goes in specific areas. We have a beaker for putting slides in, and it'll have disinfectant or bleach in the beaker, and you put the slide in there. The cover slips go in a different beaker, and the cover slips will be thrown away. The slides will be washed and then reused. Broken glass goes in the broken glass container. It never goes in the trash. Glass never goes in the trash. Glass never goes in the autoclave bag. If you have bacteria, like a paper towel that's been contaminated with bacteria, or your petri dishes that have bacteria, it should go in the autoclave bag, which is also called the biohazard bag. And that biohazard bag will be autoclaved before it is discarded, and that's to kill any microorganisms. And anything that has microorganisms on it should go in the autoclave bag or the biohazard bag. Any question about that? The exception is if you have glass, it does not go in the autoclave bag. Glass is always special and it is put in a special container, usually beakers for glass. Any question about where you're discarding materials? All right, you will be quizzed on that, so. All right, let's move on. So the schedule is given in the syllabus, the last page of the syllabus. Today we're covering Lab 00. On Thursday we'll be covering Lab 01, sorry, Lab 1. Today is going to be going over laboratory safety and training. At the end of this week, you should read the Infectious Disease Research Project. There's a file called Project Instructions on Canvas. You have to have that read for next week. You don't need to do it right away because obviously you can do it after Thursday, but it should be read before next week. I'm going to be talking about the project, the Infectious Disease Project, next week. That's why I want you to read that. Please note that the online plagiarism quiz is due at 11:59 p.m. this Saturday, so that would be April 10th. You must achieve 100% on this plagiarism quiz. If you don't have 100%, then you'll get 0 on the 100-point infectious disease project. Any question about that? You can take the online plagiarism quiz something like 5 to 7 times. So if you don't get 100%, just take it again. All right, any questions about what we're doing? Do I have people here? Hello?
Yeah, I'm here.
Yes.
Okay, good.
Yes.
I don't
here.
see any pictures here of anyone, so that's why I'm asking. Yeah, I don't have anything now. This isn't quite working right. Hopefully it's working right on your end. All right, let's go over Lab Module 0 on laboratory safety. If you didn't work safely in the lab, if you were actually going into the lab, you could be kicked out of the lab. Primary learning objectives for this lab. Upon successful completion of this laboratory, you should be able to distinguish between safe and unsafe laboratory practices. You should understand the relevant safety equipment where it's located in the lab and understand how to safely dispose of contaminated materials. So we're going to go over that again. It's not exactly important that you know where the safety equipment is, this, like the fire extinguisher, because you're not actually going to be going in the lab. All right, so laboratory safety, these rules must be followed. You must wear proper attire, which includes, when you come in, obviously you must have your clothes on and your shoes on. And the shoes have to be special, meaning they have to be a closed-toed shoes. Some women have open-toed shoes. Those are not allowed in the microbiology lab. You also have to put on a lab coat that'll cover your lap when you're sitting. You have to wear disposable gloves. And whenever there's a splash hazard, you have to wear goggles. You then take those off before you leave the lab. Generally take the goggles off first, and then the gloves, and then the lab coat. The lab coat will be hung up. The gloves are, if you got bacteria on them, you put them in the biohazard bag. If there's no bacteria on the gloves, meaning you didn't splash or spill anything on your gloves, then you can throw them in the garbage can. And then before you leave, obviously hang up your lab coat, wash your hands before you leave, and then leave. The long hair should be tied back. No food, eating, or drinking in the lab. No use of any electronic equipment. Actually, everything you own should be in your backpack and should stay there until you leave the lab. No personal items are allowed into the main part of the lab, including cell phones. And we already talked about that. So this is what you should be look like while you're in the lab. And those are eyeglasses, which are goggles, not personal eyeglasses. And we have the big goggles, which fit over eyeglasses, and most of ours are big goggles. Keep the work area clean. You should disinfect the lab station, the bench at the beginning and at the end of your lab session. That is what our lab session actually looks like. Discard waste appropriately. We don't really work with sharps, but paper towel that's contaminated goes in the biohazard bag, also called the autoclave bag. Gloves, if they're contaminated, go there. If they're not contaminated, the gloves can go in the garbage can. Paper towels that are not contaminated with bacteria can go in the garbage can. Glass goes in specific beakers, like we have one specifically for slides, another one for cover slips. We do have a beaker for, what do you call them, Q-tips, swabs. If you're working with glassware, you would have to ask me where to put it. We don't usually work with beakers and glassware, but if you are, then you put it at the back of the lab with the glass test tubes. The glass is always handled separately than all other materials. And that's because we don't want anyone getting cut on the glass. Report all spills immediately to your instructor. You should cover the blue stuff is the spill, and that'll have bacteria in it. You should put disinfectant down on the spill and then cover it with a paper towel to wick the disinfectant all over the spill, and then let it sit for 15 minutes and then wipe it up. And that should be discarded, assuming it has no glass in it, in the biohazard trash autoclave bag. No mouth pipetting, no chewing of anything in the lab. To sterilize the equipment we work with, which is a loop, we put it in a Bacto incinerator, and you wait until the metal part is, the loop part is red hot, and then you take it out and use it. You should be familiar with the safety equipment, but we're just going to show it to you here because you're not going into the lab.
I have a question for you.
Yeah.
Will we have access to this document after?
You should already have access to it. Let me look on the lab webpage.
Maybe I missed it.
Yeah, it's right there. Module 1, Ubiquity of Organisms. It's even a...
Okay. Thank you.
You already have access to it. Never take shortcuts, because that risks the safety and accuracy of what you're doing. And if you're risking the safety, you're going to bring microorganisms into the environment, which can get you and other students contaminated. So don't take shortcuts. I'm not going to talk about the deduction of points if you do something that's unsafe because you're not going into the lab. You should handle all cultures, that's a microorganism you're growing, and treat it as that it's potentially pathogenic. Now, obviously, most of the organisms you grow are not human pathogens, but some of them could be. And so you treat everything in the microbiology lab as potentially pathogenic. Keep your hands, pencils, and other materials out of your mouth, your ears, your nose, etc. No eating, drinking, smoking, gum chewing, or any other oral habit in the lab. You always wear gloves. And then if you were going into the lab, you monitor yourself for coming down with a possible bacterial infection. Closed-toed shoes is required at all times in the lab, and women can wear, I've never seen men wearing open-toed shoes, but if somebody came in with something like thongs on, they can wear that into the lab, but in the lab they must switch shoes and then wear closed-toed shoes while you're in the lab. The reason for the closed-toed shoes is that if you had something, hopefully you can see this, and then you were to accidentally spill it on your feet, the closed-toed shoes would give you minimum protection. If you had open-toed shoes, you'd have no protection and your feet would get contaminated with bacteria. All right, any question about any of that? Mouth pipetting is not allowed. No cultures are obviously ever taken out of the lab. You could bring in a sample into the lab, but only when you clear it with your instructor beforehand. For example, if someone were to say, my mother had MRSA, can I bring a sample of that into the lab? I would say, no, I don't want that in the lab. However, if you were bringing something like, a sample from your dog's mouth, I would say, okay, you could probably bring that into the lab. We already talked about discarding, so I'm not going to go over that again. All right, I think that's it for this lab. Let me explain what you're doing. And you're going to be doing a lab worksheet, which is right here. Click on the worksheet, lab, whoops, that's the wrong one, sorry. Let me go back. Lab 00 worksheet.
I was
0.5 points, what?
you said that you had already posted it on there and that it was under the ambiguity of microorganisms, correct?
Yes, it's already there.
That when I click on that link, it brings you to a different doc than the one you were just showing us.
All right, let me take a look. It might be a similar document if it's not exactly the same document.
Well, when you go through it, a lot of the information that is needed, because I was looking at it earlier, trying to do some of it before class. A lot of the information that you have on your slide isn't on there because I wasn't able to answer the questions, but now watching you go through your slide, I can see that it's different and has different.
Yeah, that's the learning objectives. Oh, this is a little different and it has an introduction. This has more material. It has the term, phrase, ubiquity of microorganisms. You should know that term, that microorganisms are essentially everywhere. And to deal with that in the microbiology lab, we give you cultures or fluid media culture, something for growing your microorganisms that's sterile, meaning it's something that's free of living cells. And how we get that is we autoclave it. I'll talk more about that later. Actually, this one is better. It has links on a video for preparing a pour plate. I didn't have that on mine. And you do need to watch that. Just a minute. So, actually, I should have gone through this one. So watch that video. And then showing you some media, that's not terribly important. I didn't talk about, there's general purpose media, and you need to know that there's different types of media. One of the types of media is a general purpose media. These are non-selective media that allow many things to grow, not all things. And they're generally cheap, and so it's generally what we use in microbiology. There's 3 general purpose media, NA, nutrient agar with yeast extract, tryptic soy agar or TSA, and NA or nutrient agar. And we use those three all the time in microbiology. You should label all plates, and I mentioned this in the syllabus, with your name, the date, the type of media, like NA, and the incubation temperature, that one is optional, and the inoculum, what you're growing in the culture. If you know what it is, write it down, E. coli, like right here. If you don't know what it is, like you're growing bacteria that you got out of the air, say something like air or air exposure. An inoculation is how you put bacteria in something and then you incubate it to grow the microorganisms in a culture media. Incubations can have different oxygen and carbon dioxide concentrations. They can grow at different temperatures, have different humidity, and obviously different time. Our incubators are simple, and we can change the temperature, and we can increase the humidity from the room, but just a little bit. And obviously the time thing. We don't have the capability of changing the oxygen and carbon dioxide concentrations. And there's a video on binary fission, how bacteria grow. We'll talk about that in the lecture later. And there's a picture of an incubator. Generally, when you're growing cultures, you can distinguish between fungus. Let me see if I can blow this up. Oop, that didn't help much. Let's go down one. Nope. There we go. Fungi are fuzzy looking and hairy looking. And then bacteria typically, not always, have a very distinct edge. And so you can distinguish between bacteria and fungus most of the time, not all the time, but 95% of the time, by the edge of the colony. If it's distinct and clear boundary, it's bacterial. If it's hairy or fuzzy looking, then it is a fungus. Any question about that? You do need to learn about the colony morphology. If I ask you questions about the colony characteristics, I will give you this slide, so that'll help you out. But you have to know how to use this slide, okay? And I won't ask you to distinguish, is that colony margin, that's the edge of the colony, is it lobate or curled or undulate? Because those are two similar to each other. But I could ask you, is it entire or is it rhizoid? Everyone clear on that? Another question I would never ask you, is that colony convex or pulvinate? Because those are too similar. I also never covered abundance and diversity. Abundance is simply the total number of colonies. Anyone want to say what the abundance of this plate is? Nobody wants to say. It's something like 30 to 50 colonies on the plate would be the abundance. I'm not going to actually count it, but it's something like 30 to 50. The diversity is the different kinds of colonies. And A is a colony, C is a colony, B is a colony, and these are all different. And then this is a different colony than A, B, and C, because it's a different color, that's a different color, that's a different color. And so the diversity would be the different colony types. And you can go by size, like this colony here is obviously different than that colony there, and that colony there, just by size and color. Okay, but you can also go by other colony characteristics by size and color. And here's one where the edge looks rhizoid, and that one the edge is distinct and clear and entire is what we call it. That one too. But these two are probably the same. Any question about that? So the abundance, you count the entire colony numbers, and I'm not going to do that because there's so many of them. Diversity is a little bit of opinion. Like some people would say, well, these three are probably the same because of the same color, but that one might be different. But some people would call them two different colonies, and others would say they're the same colony. And that's a little bit of opinion there. And so diversity is a little bit of an opinion. Any question about that? Here's a real world example. The abundance on this picture of cats is 1, 2, 3, 4, 5, 6, 7. Cats is the abundance. And then the diversity is 2. There's cheetahs and there's lions. Any question about that? You should know the terms.
Could I ask you a question?
Yeah.
So this one does have a lot more information. Although the lab 00 assignment, yeah, it's about the other document you're showing us, not the one that we have access to currently.
Let me look at that. All right, so these questions refer to the lab, what you put on and keep on yourself. That is given. I think it's even given in this worksheet. Nope, it's given here. What you wear in the lab.
Can you show us where? Because I think we need the other document shared with us. The one that you're looking at before is the one to the 00 lab, I believe.
Oh, that's the wrong one. That's lab module 1, not 00. Let me take a look at that. You know something? Lab module 0 isn't here. Now it is. My apologies.
That's okay. Thank you so much.
Supposed to be there, and for some reason it wasn't. All right, there it is there. Anyways, click on this worksheet here to get this one, and then type your answers in a document. Let me open it this way because I'm not seeing it very well. That's not the worksheet. Type your answers in here, and these questions were very confusing, so I specifically put them down here. In the lab, what do you always wear for safety before you enter the lab? After you enter the lab, what do you put on to keep yourself safe in the lab? Each chemical in the lab must have a safety data sheet that describes what? And this one, you only need 2 things, so the asterisks tell you about how many answers you should have. And rule 3 is 1, 2, 3, 4, 5 different things that I'm looking for. Know the location of your safety equipment, such as a fire extinguisher, a fire blanket, an eye wash station. What can you take into the lab? And can you eat and drink in the lab? So that should be pretty easy. List one item of laboratory attire you must wear in a microbiology lab. List one item that is allowed in a microbiology lab. So what can you take into the microbiology lab? Some people were saying nothing. That's not entirely correct. For example, if you have eyeglasses, you can take that into the microbiology lab. And you wear them while you're in the lab, and you can take them home with you. You know what I'm saying? Everyone get that? There are some things that you are allowed to take into the lab and then take out with you. But don't take the glasses off and put them on the counter. Okay? Keep them on your face. But you should not take your cell phone into the lab and then put it down on the counter. You should not take a pencil into the lab, put it down on the counter, and then take it home for you. If you wanted a special pencil or pen, you can bring it into the lab and then just leave it into the lab. Leave it in the lab and it can stay there. Then list one item that's not allowed in the microbiology lab, and then submit this file by, actually it says 8 a.m. Friday of week 1, but actually I think you can submit it until Saturday in week 1 in the worksheet lab 00. Okay, and you must submit lab 00 worksheet, oh that's why I said by Friday, because by Friday I've got to figure out who's in the class, and if you don't have it submitted by Friday, I won't know who is in the class and who's not in the class and who's come, and so I'm using this to take attendance and figure out who's actually showing up. So you must submit this by 8 a.m. Friday of week 1. I'm going to change the due date because that's because I need to take attendance, and anyone who doesn't have it in by 8 a.m. on Friday, you're going to be dropped from the class, because I have to do by Friday at 5 p.m. I have to drop any student who's not shown up for the class. Okay? Any question about any of that? All right, go ahead and get started. I will be here until 8 o'clock to answer any questions.
Thank you.
All right. I'm changing the due date.
Thank you. Have a good night.
Yeah, you too.
Thank you.
Thank you.
You're welcome. Ah,